Okay, so nerve stains. Basically, yeah, we are talking some really easy stains, and then we're talking silvers. Okay. And some of your silvers you're gonna find, yeah, really it's up to the pathologist whether they want to counter or whether they don't want to counter. Yeah. And that's with those silver stains. But again, Carson's going to tell you to counter with different things, like aniline blue, light green, NFR. Yeah, and that's what you need to know what you can and cannot counter with. But always keep in mind that it's really up to your pathologist because we're talking about nerves here, right? We're talking about neurons. Yeah, and so sometimes they don't want you to counter because they don't want you to overlay. Okay, but we're going to start with a review, and then we're going to talk about the easy stains, and then we're going to get into some of the harder stains, the silver stains, and then we're going to look at some really cool 3D mapping. So, as always, you're you are, going after those steps, functions, and results and controls. Yeah, controls are pretty straightforward. Yeah, with this chapter, I mean, it's nerves, so 90% of the time you're going to be using brain, right? Yeah. So steps, functions, results, and controls. Yeah, so here we go. Why am I not going anywhere? I'm not going anywhere. Let's see what's happening. Oh no. Um, okay. Hold on here just a second. Let me figure this out. Okay. Okay. So here we go. Steps, functions, results with a little review of your nervous system first. Yeah, so you've got two parts to your nervous system. We got the CNS and we got the PNS. Yeah, the CNS is your central nervous system, PNS is your peripheral nervous system. Now, as far as neurons go, they are composed of three independent parts. The first part is the cell body, and that's the main portion, the cellular portion. Yeah, then we have dendrites. So, dendrites conduct impulses to the cell body. They don't have myelin sheaths. They're usually short, highly branched, as opposed to axons. Axons conduct impulses away from the cell body. So, I always like to do, sort of this thing and go, okay, if my head, is the cell body, then this little short guy right here, this is your dendrite. Yeah, and he is sort of speaking to an axon on this side, and they're interchanging information. So your axon is reaching away from your cell body and it's transferring information to the next neuron, but it's not transferring to another axon, but rather to another dendrite that then is pulling that information into the cell body. Each neuron only has one axon, and it originates from that cone shape, of the neuron itself. So if you look at this guy up here, you can see how they're sort of triangular shaped cell bodies. And you will never see, another cell that has that triangular shape. But you have to keep in mind, the way that the cell is actually cut and laying on the plane of your cut. So you may see it this way, or you may see it this way, or you may see it this way, or you may see like half of it. Just depends on how you get that cut on that neuron. So just a, sort of picture of how they're transporting information. So here's your cell body. These dendrites, there's multiple of them bringing information into the cell. Yeah, and you can see they're highly branched. Here is your axon reaching out to the next cell, and your axon is communicating with the little highly branched dendrites. So they're classified into three classifications. There's sensory neurons, motor neurons, and interneurons. Yeah, sensory neurons, they conduct impulses to the spinal cord and to the brain. So that's information going in. Yeah, motor neurons conduct impulses away from the brain and the spinal cord. And then interneurons conduct impulses from the sensory neurons to the motor neurons. So, I'm sending information to your brain. Yeah, here, it's kind of an illustration. Here's our brain. Ooh, ow, we stuck our finger in the fire. You pull your hand back. Ouch, that hurt. And then your hand automatically, your brain automatically goes, ooh, you know. So you're like, oh, fire, oh, take your hand away. Yeah, it's really kind of a simple concept. Yeah, so your motor neurons are sending a message to your brain, and to the muscles in order for them to move that hand away from that flame. Your interneurons are the little guys that have synaptic endings, and they are the ones who are sort of in the middle, in between, your dendrites and your axons. They're interactive cells, or interneuron cells. You'll see some of the old literature call them interactive cells. So, Nissl substance. Nissl substance is a basophilic material. It is let me put this over here. There we go. It's a material that uh, is in large aggregations of rough endoplasmic reticulum. And it does have RNA content. Yeah. And because of that RNA content, the Nissl substance will stain with basic dyes. Now, if you have a loss of Nissl substance, we are talking about a disease that's called, I always mispronounce this one, chromatolysis. Um, and so that's a degradation of your endoplasmic reticulum and RNA content within your brain. Now, it's sort of weird when you look at Nissl substance, yeah, microscopically, because Nissl is actually intracellular, so it's inside of those neurons. But microscopically, it can look like it's extracellular because it's all about that cut, right? So if you have a neuron that's triangular shaped, yeah, and you get a cut that's, yeah, you can't see the whole wall of that neuron, and you get a cut that is, say, half of that triangular shape, that Nissl substance can appear to be extracellular. And it can potentially fool you, but just keep in mind that it really is intracellular and, excuse me, it's intracellular, and that's just the angle of the cut that makes it look extracellular. So this is what it looks like. All of this granulation, here's your neuron. Here's a dendrite. Here's a dendrite. This is your axon coming out. Here's your nucleolus. All this purple granulation in here is Nissl substance. So then we have glia cells. Glia cells are referred to as nerve glue. Yeah, they are also referred to as neuroglia. So these surround the neurons. And so they supply nutrients, they insulate the neurons from the neurons bumping into each other. Yeah, they destroy pathogens, they destroy the dead neurons that are in your brain, and they regulate that sort of internal environment of your brain. If you think about, you think about the brain when it's unfixed and it's sort of jelly-like. Yeah, that jelly-like substance is a conglomeration of glia cells. Um, so they're your glial cells. Surround those neurons and they create this sort of support structure, much like scaffolds that hold everything up. And they are comprised of different types of cells. So glial cells or neuroglia cells um, aren't a specific type of cell, but rather a conglomeration of cells that include astrocytes, microglia, ependymal cells, and oligodendroglial cells. So, yeah, astrocytes, these guys sort of wrap themselves around the myelin sheath. Oligodendroglia attach themselves to those sheath bodies, and microglia are sort of free out there roaming around, as well as your ependymal cells. All right, so oligodendroglia. Oligodendroglia are the maintenance men of the brain. So they're small cells that are in the central nervous system. Yeah, and their whole thing is to maintain the myelin sheath and the axons. Yeah, so they are sort of the cleanup crew, the maintenance men. They fix anything that goes wrong with, yeah, your myelin sheaths or your axons. Yeah, they're the most numerous within the glial cells, and they're found both in the white matter and in the gray matter. So, if we look at the myelin sheath, so here's, myelin, right? Yeah, I mean, each one of these is a segment of myelin. In between the two is what's called the node Ravier. And so what this uh, oligodendrocyte is going to do is it's got little arms on it, and it's going to wrap itself around a myelin sheath. Each one of those arms is going to wrap itself around a myelin sheath, and it's just going to run up and down that one little stretch. Yeah, and it's just going to constantly maintain that myelin. Then we have our astrocytes. So astrocytes are sort of anchors, if you look at it this way. They anchor the small blood vessels to the neurons to feed the neurons. They're stellate, which means that they're star-shaped. And what they do is they function in scar formation and injury. Um, So they provide support, and they also participate in the exchanges of gases and the exchanges of metabolites. So, stellate is a term that you're going to see used often, not only in terms of astrocytes, but also in terms of tumors. Yeah, whenever anything is star-shaped and finger-like, yeah, stellate is a good terminology to use. So here's just a picture of an astrocyte, and you can see how he's reaching out and participating in the exchanges of gases inside that nota raviere. Okay. So here we have, two sheaths, yeah, of myelin, and the nota raviere has all the exchanges, chemical and biological exchanges happening, and the astrocyte reaches out and sort of participates by injecting its little tentacle, if you will, around that nota raviere, and engaging in keeping those gases, yeah, within the structure itself. Then we have microglia. So microglia, these are your phagocytes, yeah, of the brain. They are your cleanup crew, your garbage men. They can be resting or they can be migratory. Now, you're going to see the term fixed, yeah, in relationship to microglia. And we are not talking about fixed as in formalin fixation. What we're talking about, yeah, when they say they're fixed, is that they do not leave the brain. Okay, so they are fixed within the brain. Yeah, but yet they are phagocytes, so they do move about. So just keep in mind, yeah, okay, they can move around and clean up, like any phagocyte can do, but microglia are specific phagocytes to the brain and they do not leave the brain. So ALS is a um, immune cell disturbance within the nervous system. And that is when the microglia cells get activated by that disease, even though there's nothing for those microglia to actually attack, destroy, and clean up. So they get activated and they actually start cleaning up and phagocytizing good, healthy cells. And they get activated by a inflammatory signal that tells them that something is wrong, but yet there isn't anything wrong. So this is sort of how it works. Here we have a neuron, and this is normal. Okay, so here we have a neuron. And these are just resting microglial cells. They're just hanging out and they are waiting for some sort of signal. Okay. They're just do, let us know when you need us. Yeah. And then all of a sudden, an inflammatory response starts to happen. And these resting microglial cells activate. And so when they activate, yeah, then all of a sudden the full activation starts to happen so that the inflammatory response signals them to, hey, wake up, we need you. And then once they wake up, they go into full activation and they start phagocytizing all of the dead debris um, and cleaning up the workspace, so to speak. However, yeah, in an ALS environment, there isn't anything to clean up. So when they get activated from an inflammatory signal, they actually, instead of destroying the, the cleanup, the dead cells, the, debris, if you will, they start attacking the actual neuron itself and start phagocytizing the neurons. Okay, so then we have the ependymal cells. Yeah, these are the producers. They line the ventricles of your spinal canal and they secrete CSF. And cilia helps move that CSF around in your central nervous system. CSF is cerebral spinal fluid. So they are constantly producing your cerebral spinal fluid for you. And they are moving and they do not rest and they line the ventricles of your brain. Then we have myelin. So myelin is your protectors, it's your armor, yeah, of your axons of your neurons. So it's a complex, white, fatty, non-living material. So in other words, myelin in and of itself does not have any um, nucleus. It doesn't have any DNA, RNA. And the truth of the matter is the myelin, when we process, um, the myelin gets lost. And what's left behind is a substance that's called neurokeratin, because the neurokeratin is part of that non-living material. So we can attach to the neurokeratin, yeah, and it looks just like myelin, yeah, microscopically, but keep in mind that the myelin itself actually goes away and we're staining the leftover, which is neurokeratin. Then we have Schwann cells. So these are your architects. They build the sheaths of the myelin that's around the axons. So just sort of a quickie here. So here's your true axon in the middle. Then you have layers and layers and layers of myelin. Your Schwann cell lives in that myelin, okay? And this is its nucleus, and this big wrap right here is the Schwann cell itself. This dark blue is the myelin wrapped around the axon. So then you have one whole sheath here a nota raviere. The nota raviere is where all the gases and things pass through. And that so this is a close-up. Yeah, so here you have your neuron, your dendrites, and your axon. Um, so your axon runs through the layers of myelin. And then at the end, it has its little communicating claw, if you will. All right, so there's a lot of nerve stains, and we have the Kreselec violet, we have the Bodian, the Holmes, the Wiles, the Thioflavin S, the Bielchowski, the Biel PAS, the Biel microwave, the Biel Munger, the Holzer, the Cahals, which you already know, the Luxelfast blue, also known as LFB, and then we have a combo Luxelfast blue CEV, which is a combination stain of Luxel Fast Blue for myelin and the Kreselec Violet for Nissl. So we're going to start easy, and we're going to start with the Kreselec Violet. And so keep in mind that there are two chemicals that sound very familiar to one another. There's the Kreselec violet and there's the crystal violet. So, yeah, we are trained to refer to the Cresol violet instead of saying, hey, grab me the Cresol violet. That can be confused with, hey, grab me the crystal violet. So we refer to the Cresol violet as CEV, Kreselec violet. So this is for Nissl substance. And chromatolysis, loss of Nissl is chromatolysis. Yeah, and chromatolysis occurs when exhaustion of a cell happens due to injury or damage. And once repaired by protein synthesis, the Nissl substance can return to normal. Yeah. So the reason it can return to normal is because it's endoplasmic reticulum. Yeah, and endoplasmic reticulum is constantly reforming itself. So you can actually come back from uh, damage or protein synthesis via protein synthesis of your Nissl substance. It's basophilic. Yeah, and there are two protocols. There's Luna and then there's Veka. Now, Veka is the easier of the two, but you need to know both. So easy, cheesy. You're going to depair and hydrate, go into crystal violet, DCM. That's it. And so your Nissl substance is going to be blue to purple, and your background will be colorless to purple, depending upon your differentiation time. And your differentiation time is in that 95% alcohol. This is what it looks like. So yeah, you can see this really nice granulation factor inside of the neuron. And that's what we're looking for is this granulation. That is Nissl substance. And remember when I said it depends on how your cut is, the Nissl substance is in fact intracellular in the neuron. But if you look up here, it looks like it's extracellular, right? And that's just because of the cut of this neuron is not in the exact plane. Okay. Yeah, somebody look in their book and tell me you should have both Luna and Veka in there. Let me let me get mine here. Let's go back.
It's listed as method 1 and method 2,
Oh, is
I
it?
think.
All right. Let me.
Yeah.
Ah, okay. So Luna, yeah, the difference between Vega and Luna, yeah, is that there's balsam in one of them. Yeah, yeah. So Luna has balsam oil in it, a mixture of balsam xylene. Yeah, and so here's how I always explain this. Your grandma made the best ham ever for Thanksgiving. And you notice that her ham, she always cut the end off of the ham and put it in the pan and then baked it. And then years later, yeah, you ask your grandma and you say, Grandma, you always have the best ham. Why is it that you cut the ends off of the ham? And, how does that make it better? And your grandma smiles back at you and says, Honey, I cut the ham off of, or cut the ends off of my ham so that it will fit in the pan. And that's sort of the difference between Luna and Vega is that for years we thought we had to protect this Nissl substance, and keep it in the section by treating it with balsam oil, yeah, before we differentiated in that alcohol. Turns out, years later, that we don't have to do that. So we don't have to cut the ends off the ham. Okay. So then Veka is the one who figured that out and made it much easier to just have the, D-parenhydrate, Cruselec violet, and DCM. So you do not, in fact, have to cut the ends off the ham. Yeah, you can achieve the same thing, without using the balsam oil. So then we have Luxel Fast Blue. Yeah, so this is referred to as LFB. And so as your axons degenerate and start to die, that myelin sheath starts to break down. And so we need to sort of look and see if that's the case, in terms of, and let's be frank, when we're talking about brain, we are talking about autopsies. We are either doing uh um, research on, animals, yeah, such as, cancer research on rats and things, or we are dealing with an autopsy case. We don't just pull out a part of a person's brain. So with the LFB, your gray matter and your white matter need to be distinguishable macroscopically. You need to be able to see that with your eyes when you are running the stain. So in the differentiation step, you have to keep diffing until the gray matter is almost colorless and the white matter remains blue. Yeah, this is an overnight stain. Yeah, so when you guys get to doing it, make sure that everybody goes together. Yeah, and make sure that your Copeland jar lid is covered, to prevent evaporation. Now, Carson runs through the diff process twice, so don't let her confuse you. If run through that differentiation process the first time and it looks good, by all means move on. You do not have to do it twice. Now, LFB, the stain itself, is often combined with other stains. So we have an LFB with a CEV tag, and then we have an LFB with a PASH tag, and then we have an LFB with a Holmes silver nitrate tag. So the LFB protocol, yeah, you're going to depair and hydrate to 95. Don't hit it with water. We have to keep that alcoholic environment. Then you're going to go into Luxil Fast Blue and Copeland jar of Luxil Fast Blue, just drop it overnight. That's going to stain your myelin. More specifically, it's going to stain the neurokeratin because remember that the myelin goes away, when we process. So then you're going to come out of LFB, and you're going to dip it quickly, in 95 to remove any excess stain off of your slide. Then you're going to water wash it, to remove that alcohol. Then you're going to go into a lithium carbonate. This lithium carbonate is a macroscopic differentiation. So once you go into this lithium carbonate, you're going to see sort of that background start to disappear. Then you're going to go into a 70% alcohol. That 70% alcohol is actually, it's fine tuning is what it is. You have to stay in that 70% alcohol until the white and the gray matter macroscopically with your eyes, are visually apparent. The two of them are visually apparent against each other. Then you water wash. Now, Carson's going to tell you from here to go all the way back to here and do it again. You really do not have to do that. The only time that you're going to have to do that, is if you look at this microscopically in your water and you find that you still have sort of a bleeding happening in between your gray and your white matter, then you can go back and fix it. But you don't have to go back and fix it, okay? Then you're going to DCM. And this is what it looks like. So you can see, how the myelin is this deep blue color, and then everything else is somewhat colorless. So, the combination stain, the LFB CEV combination stain, the CEV is going to just simply intensify that LFB staining. So in order to achieve that, we are going to depair and hydrate to 95, go into our Luxil Fast Blue overnight. We're going to come out, remove excess. It's basically an LFB, okay? You're going to come out to remove that excess, water wash, do your first differentiation, and then your fine tuning differentiation and water wash. Then you're just going to tag it with a crystal violet. So Cresol violet, CEV. So you're going to go into your Cresolect violet. That's going to stain your Nissl substance. Then you're going to differentiate that Cresol violet with a 95. Then you're going to go into 100 and clear and mount. And this is what it looks like. So your myelin is this blue striping, little reaching axons everywhere. And then you're going to see your neuron cell body and its Nissl. Then we have the Wiles stain. Um, for myelin sheath. The Wiles isn't used as often, um, because we can do the same thing with the LFB, but it is still out there. So your the mordant is the hematoxylin solution attaching to that myelin sheath. Then you're going to do a regressive staining. So you have two diff stains. So your first differentiation is macroscopic, and it's done with ferric ammonium. Yeah, and so that removes any excess mordant that may be there. Then your second differentiation is microscopic, and it's done with borax fer-I. Notice that fer-I, it is bolded. This is not ferrocyanide. This is fer-I cyanide. And if you're not paying attention, you can actually use a ferrocyanide when it's supposed to be a fer-I cyanide, and they are two completely different animals. So you've got to really pay attention to make sure you have the right solution there. Excuse me. So, yeah, it's going to remove any non-bound, unbound, non-specific hematoxylin lake, and it forms a colorless oxidation product. So we're going to depair and hydrate. We're going to go into our hematoxylin lake. This is going to stain our myelin sheath. Then we're going to water wash to remove any excess hematoxylin. You're going to go into your first differentiation, which is ferric ammonium sulfate. And this is when you're going to see with your eyes macroscopically that gray and white matter become very distinguishable against itself. Yeah, then you're going to stop that water wash with a differentiation. Then you're going to go into sodium borate, potassium ferricyanide. This is your second differentiation, and this is your microscopic differentiation. So you want to pull out of your second, go into your water wash to stop that second differentiation. Take a look at it microscopically. Okay. If it's not very sharp detailed between the gray and the white matter, then you can go back into your potassium ferricyanide. Then we're going to go into a dilute ammonia water. That ammonia is going to lock in that mordant lake. Then you're going to water wash to remove the ammonia and DCM. And this is what it looks like. This is actually a full crosscut of a rat brain. So your myelin sheaths are here in this blue, dark blue area with the hematoxylin mordant lake. And then your background is tan. Yeah, this is what you're differentiating out. Okay. All right, so we're going to get a little bit harder now. So the Bodian method for nerve fibers, nerve endings, and neurofibrils. Yeah, so this guy uses copper. Yeah, so copper is added to the impregnation solution. And what that copper does is it destains any connective tissues, yeah, so that we are not staining connective tissues that wind up being big blobs, if you will. Yeah, we destain them so that we don't see them. Yeah, it's hydroquinone is used as a reducing substance, and hydroquinone is that carcinogen. We're going to tone with gold. Oxalic acid is used to intensify and bring up that stain level. Yeah, then we use hypo, our friend, And counter staining, again, up to your pathologist, but the most common is aniline blue. However, if you think this is a silver stain, obviously, so if you think about your nerves, your neurons, and your dendrites being black, yeah, and then having a blue background, and aniline blue at that, which can lay over. Sometimes it's difficult to distinguish that blue from that black, especially if you've laid over. So, NFR can also be used. So, our protocol. This is a silver, so we're talking distilled water. Yeah, so we're going to deparenhydrate to distilled. We're going to go into what's called a protargal solution with a copper shot. Yeah, so this is an impregnation step, and it takes 48 hours, 2 days, to complete. And remember, this copper shot is going to remove the coloration or prevent the coloration of your connective tissues, while the protargal solution is a silver solution. Yeah, that is going to impregnate your neurons. Yeah. So we're going to come out 48 hours later. We're going to distill water wash. We're going to go into a reducing solution, which makes this our gyrophilic. Yeah, and it's hydroquinone with a formaldehyde solution. Then we're going to distill water wash. We're going to tone with gold chloride. We're going to distill water wash. Then we're going to go into oxalic acid. Now, this oxalic acid is going to reduce the gold coloration, and intensify the stain. So this is a developing step, okay? When you, it's going to be, your silver is actually going to be really goldish kind of color from your protargal. And yeah, so we're going to reduce that gold down to a metallic black. Then, yeah, we are going to water wash our friend. We're going to remove any excess protargal solution that is not bound. Then we're going to distill water wash, and we are going to counter stain. Now, again, it's another situation where it could be aniline blue or NFR, or no counter stain at all. Then you're going to dehydrate, clear, and mount. So your results, your new nerve fibers are going to be black, and that's from the protargal. Your background will be light blue to gray. And that's from you um, differentiating out the hematoxylin solution, the protargal. Your nuclei are going to be black. Your background will be blue, or red, depending upon whether you've used aniline blue or NFR. Okay. So we have an interesting, so this is what it looks like here. Okay. So this is really cool. We, a lot of our brain research was done on crawfish, or sea mantis, whichever you prefer to call it. Turns out that the sea mantis or crawfish, its brain is very much like our brain in terms of how it reacts to the silver solutions, in terms of how it stains. And you can see this is actually a crawfish. So you can sort of see those nerve fibers running. And they're nice and black. They're actually quite big compared to ours, but we can get a full cross cut of a crawfish. And all of the studies, in terms of developing brain stains were done on our crawfish. So then we have the Holmes. The Holmes is a modification of the Bodian method. Okay, that's all it is. It's a derivative. Holmes developed a buffered impregnation solution. And it increases the alkalinity. So, what he did was he added pyridine, to the solution. And that's really the only difference between the two, is sort of, yeah, buffering out that impregnation solution, so that we increase the alkalinity inside of our stain. So we're going to depair and hydrate. We're going to go into a 20% silver nitrate. That's going to sensitize. So you're using a diamine silver here in um a, 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 a in a stain to sensitize for your ammoniacal silver. Okay, so 20% silver nitrate, distilled water wash. That's just going to remove that extra silver nitrate. Then you're going to go into a second impregnation. Yeah, so that's your silver nitrate plus your pyridine. This is your change here. And this is overnight. Then you're not going to wash, but rather, you know how when you come out of your water bath and you flick your slide to get that excess water off? That's basically what you're going to do here, although flick it onto a chem wipe. Okay. Yeah. Then you're going to go into your reducer, yeah, which is hydroquinone based, not formaldehyde based. Yeah, then you're going to distilled water wash, and then the typical. Yeah, you're going to tone with gold chloride, distilled water wash, go into your oxalic acid. Yeah, distilled water wash, our friend hypo, water wash, and then counter stain if necessary. So up here is where the change comes, in between the Holmes and the Bodian. So your results, your axons and nerve fibers are going to be black. Your nerve fibrils will be black. Your background will be light blue, light green, or red, depending upon what your pathologists want. And it looks identical to the Bodian. Then we have the thioflavin S. Now, this is not like the thioflavin T. The thioflavin T, um, it's not affected by prolonged fixation like the thioflavin T is. The thioflavin S is going to stain both amyloid and neurofibrillary tangles and plaques. So neurofibrillary tangles are an indicator of Alzheimer's disease. Yeah, and I'll show you what that looks like here in a second. And years and years ago, yeah, before the invention of charged slides. Yeah, we used to put egg albumin, yeah, which was powdered egg whites, in our water bath, and that would help adhere the section to the uncoated slide. We don't do that anymore. But Carson does make issue to state that you should not use any adhesives in your water bath. But quite frankly, you shouldn't be using adhesives in your water bath anyway. Okay. So our protocol for the thioflavin S. It's a thioflavin, so it's a fluorescent stain. So you're going to depair and hydrate to water, give it a nice good rinse. Carson suggests that you hold in that water for at least 5 minutes. And the reason for that is because you have to make sure every last little tidbit, of alcohol is gone. If it is not gone, it's going to interfere with the entire process and you're not going to fluoresce microscopically. So then you're going to go into potassium permanganate. That's our oxidation. Yeah, and remember that potassium permanganate is that deep, rich, purpley red color. So when you water wash afterwards, you have to make sure that all that excess pot perm is gone. Then you're going to go into metabiosulfite that has oxalic acid in it. This is going to bleach your section. Yeah, remember that pot perm is that deep purple, so we got to bleach out those striations. I shouldn't call them striations. We need to bleach out that background. Then you're going to water wash. That's going to remove any of the metabiosulfite or the oxalic acid that's attached to the potassium permanganate. Then you're going to go into sodium hydroxide or peroxidized solution. Yeah, and that's going to remove any lipid fluorescence that is there. We don't want the lipids to show up. We're even though the lipid themselves are gone through processing, but the cell wall is still there. Those cell walls will fluoresce. So we need to block those from fluorescing. Then we're going to water wash to remove that hydroxide or peroxide. Then we're going to lock it in, yeah, with that acetic acid. That's always our friend. That's always our lock. Yeah. Then you're going to water wash to remove any excess acetic acid. Go into 50% alcohol. Now, yeah, this is because that thioflavin is alcohol based. Okay. And that's the whole reason why we had to get rid of all the alcohol here, because throughout this, we're going to have an attachment to that alcohol. And if we remain with alcohol, yeah, on our slide, then what's going to happen is once we hit here, then everything that has alcohol is going to fluoresce. So your 50% alcohol, you're going to go into 2 changes, yeah, and that is going to diff that thioflavin and begin your dehydration. So here we're going to introduce the alcohol. We are going to stain with our fluorescence. Then we're going to diff with the 50% alcohol. And then these alcohols in your DCM are going to pull any of that excess water out, and so that we can fluoresce. Yeah, so neurofibrillary tangles, plaques, and amyloid are all going to be bright green. So all of these that you see, this is your fluorescence. This right here is a neurofibrillary tangle. And these are little tiny nerve fibers, yeah, that are wrapped around what's called a Purkinje cell. And I think that's what's coming next. Yes, is the Bielchowski. So, you've heard me talk about Bielchowski. Yeah, amazing guy. His first name was Max. He's German, extremely impressive guy. He published more than 180 medical articles um, related to histology. And it says that, if you do research on him, it says that he was a quiet, diligent, intelligent man. So he's quite the influencer, if you will. Yeah. in histology. So the Bielchowski-Munger. This is the easiest and most reliable of the Bielchowski stains. So we're going to depair, it's a silver, so we're going to depair and hydrate to distilled water. We're going to preheat a 20% silver to 60 degrees, and then we're going to add our slides. So again, this is a water bath thing. So you put your 20% into a water bath, heat it up, and then add your slides to it. So this is an impregnation and a sensitization step. It basically, the truth of the matter is it's used as a sensitization because it's a diamine with an ammoniacal silver. But if you were to pull your slides at this point and take a look at them, you would see that some of your axons are actually going to be impregnated. So that's why they say impregnation and sensitization. Yeah, then you're going to distilled water wash and go into your ammoniacal silver. This is your true depth of impregnation. Then you're going to water wash, hit it with hypo, water wash, and DCM. This is the one you guys are going to do in the lab. This is what it looks like. So neurofibrillary tangles. These are Purkinje cells. This is a Purkinje cell, and this is a Purkinje cell. The Purkinje cells line in between your gray matter and your white matter. They are the cells that give you time and space. Yeah, meaning they tell you where you've been, what you're doing, where you're going, and where you are. Yeah. So once the axons, the neurofibers start tangling up around a Purkinje cell and squeezing off that Purkinje, and then the next one gets squeezed off. This one's starting to be squeezed off here. You can see that the cellular material is starting to degrade. These guys have already been squeezed to death. Okay. So once you get all these neurofibrillary tangles and your Purkinje cells get squeezed off, you no longer have the ability to discern, where you were, where you are, where you're going. Hence, you have Alzheimer's disease. Now, there's variations of the Biel. So there's a Bielchowski that's tagged with a PAS. There's a Bielchowski that's tagged with a Congo red. And then there's a Bielchowski that's tagged with the LFB. Then there's the original Bielchowski. Yeah, and this original Bielchowski follows those exact steps of that ammoniacal silver. So you have way more steps to it than this does. That's why this is the easiest one, and it's also the most reliable one. So these are, oops, I'm going the wrong way. These are ones for you to take a look at and study on your own. They're pretty straightforward. You're just tagging stains that you already know onto the end. This is what a Biel Congo red looks like. So here's your Bielchowski. Yeah, and so you can see all of the nerve fibers, the axons running through there. Then you have a hematoxylin, and you have the Congo red. The Congo red is actually staining what's known as brain amyloid plaques. And amyloid plaques shows up in the brain along with Alzheimer's. So once those Purkinjes get squeezed off and they die for whatever reason that we don't know why, all of a sudden plaques starts to show up in those areas. And that plaques is actually amyloid. Then this is a Biel LFB. So you can see the Beal stain in terms of here are all your axons running through it. And then you can see the LFB staining myelin sheaths. This is the original Beal. Um, the original Beal is much darker. Yeah, it's harder to see and it's not as reliable as the um as your Beal Munger. Yeah, your Beal Munger. So Bielchowski developed the first one. Then Beal and Munger got together and they modified his original one to make it easier. The original Beal doesn't really darken the neurons, the actual axons of the neurons. Yeah, they sort of remain, you can sort of see right here, they remain sort of a brown color, as opposed to a dark black color. So that Munger is much easier. Now we got some cool stuff to look at. These are 3D mappings of the brain. Yeah, so they're, here, like here's the neuron, and you can see all of its little dendrites coming out. Here's a neuron, here's its cell nucleus, and here's a axon running to the dendrites and all the cells. Here's another axon. Here's two talking to each other. It's very cool. If you are interested in seeing more 3D mapping, there's the website for you. This is more. So it kind of gives you a look into how very far and how very long your axons can be. When you look at that, the Bielchowski, the VVG and the Bielchowski are my favorite stains. The VVG because it's fun to run. The Bielchowski because I get lost in it, even though microscopically, even though it's just brown and black. But the reason I get lost is because I can find um, a neuron and then find its axon and then follow it and just keep following it and just keep following it until you find who it's talking to. Sometimes that uh, axon, yeah, will stretch so far. It's amazing microscopically. It's amazing to see how far they can stretch to communicate to another cell. And then here's, another mapping and another mapping. So here we have that cell body. Here is, yeah, its neuron. Here you can see, little dendrites sticking out. This one's talking to this one. You can even see a little bit of exchange of gases happening. It's very cool. And I like this because it looks very much looks very much like this. The end. And